My name is Vasileios Stathias, and I'm a lead data scientist here at Sylvester Comprehensive Cancer Center and at the Department of Pharmacology at the University of Miami. My work involves leading the development of of a data platform for the Cancer Center to manage, analyze all the next generation sequencing and biomedical data that we generate here, and also to integrate various machine learnings and bioinformatics approaches to discover new drugs and compounds. My talk has two parts. On the first part of my talk, I'm gonna give you a brief description and go over our newly developed data platform here at the Cancer Center, and that gives the ability to the researchers to have fair data and also explore intuitively their data sets. On the second part of the talk, I'm going to give you a few examples of how now when we have the well annotated and curated data sets, we can then leverage different machine learning and deep learning approaches and other bioinformatic approaches to discover and make predictions for new drugs, new biomarkers, and new therapies. So this is the two parts of my talk, and we can start with a brief introduction about cancer. As all of us know, cancer is a major public health and socioeconomic issue, and its burden is set to increase by 60% by year 2040. As in many countries, in most actually countries in the world, cancer ranks as the second cause of death, right after cardiovascular diseases. And on the global level, every year we have more than 18 million cases and 9 million cancer-related deaths. During our lifetime, 21% of males and 18% of females are going to develop cancer, and 13% and 9% respectively are going to die from cancer. So overall, cancer is a major issue that impacts across our society, and we need to immediately address it. Over the past 40 years, of course, there's been a tremendous improvements in both the early detection and in the treatments of cancer patients. As you can see here in the top of the figure, for cancer types such as prostate cancer, thyroid, and skin cancer, since 1970s, there's been a huge improvement in the survival of their patients with using new treatments and early detection methods. However, on the bottom of the graph, you can see that for cancer types such as lung cancer, liver cancer, and pancreatic cancer, they have seen very limited survival increase of the patients. So there is an immediate need to find new therapies and or new detection methods for those cancer types. And in my lab, we are very interested in particular specialized in glioblastoma, which is an aggressive brain tumor. And you can see that still year 2021, the 5-year survival rate is less than 10%. And to put into perspective, this is the therapeutic landscape of glioblastoma for the past 20 years. There's been only one major therapeutic breakthrough, which is the temozolomide treatment that was introduced in 2005, that only increased the survival of the patients by a few months, and followed by both the bevacizumab and the tumor treating fields, which again did not have a substantial increase in the patient survival. So in glioblastoma and other cancer types, there is a need to find new methods and new ways to process our data and our biomedical information to discover new targets and new therapies. On the positive side, we are in a time where new technologies are rapidly evolving and are enabling new therapeutic approaches. So apart from the traditional mutational data that we saw when they were doing the drug discovery phases in the past years, we now have available transcriptomic data, proteomic, imaging, metabolomic, and epigenomic data for the tumor. And that can give us a different view of the tumor biology. Moreover, these technologies, apart from those different data types, the technologies improve on the resolution as well. Like a few years ago, it would have been unprecedented that we could look on the single cell level, both the genomic and the transcriptomic and the epigenomics of cancer cells. And of course, everything goes by with the increase of computational power that is really helping our analytical capabilities. So overall, all of these, both the different data types and the increase in resolution, can lead us now to find better therapies using precision medicine approaches where you define the right drug for only a specific subpopulation of the patients, drug combinations, which is a field our lab is also very interested, which is trying to find concurrently two drugs that are gonna be effective against the specific tumor. Computational drug design is very important, and it's been helping with both the computational increase and the new biochemical data. So design better drugs with higher efficacy. And lastly, drug repurposing. It's a major field that we can use now all the existing data and repurpose already approved drugs to go from an already approved tissue type to a different tumor type. In addition to all of those multi-omic data, we have another advantage at our hand, that the cost of generating new data is decreasing. As you remember, in 2001, it costed about $100 million to sequence the first human genome. And now we're seeing prices definitely less than $1,000, sometimes less than $200 to sequence a human genome. With this decrease in pricing, of course, has led to the increase of the overall data that have been generated. It's been projected by 2025, we're going to have about 40 exabytes of data to be stored only for human genomes, not mouse genomes or other genomes. So we have both more new technologies, we have cheaper technologies, and also we have a large generation of data nowadays. And all of this unprecedented amount of data that's been generated creates this biomedical big data ecosystem where we have all of those different technologies and different data types. And here I can highlight a few that we can use for actionable insights down the road. Then the major one, the big one, is the multiomics profiles, which we use now the technologies that we mentioned before on the DNA level, the RNA, the protein level, and profile tumor types, tumor cells, and other cell types. Another category of data, which our lab in the LINX consortium was mostly working on, is the perturbation response profiles, which are extremely valuable because they tell us what happens. We're not interested on the baseline profile of the cell or the tumor, but we're interested how the cell reacts to a small molecule treatment or a CRISPR reagent, and on the RNA level, on the protein level. So what changes happen on the transcriptome of the cell after it gets treated by a compound? Typical data for cheminformatics are the bioactivity profiles, and now we have higher and large-scale screenings to give us the association between proteins and compounds. And of course, drug and gene sensitivity profiles are invaluable. So here we want to see if the cells essentially are sensitive to particular drug treatments or gene knockdowns. Imaging data, especially in AI, are very, very important. And we can have both the large image scale data from radiology, but also cell-based imaging data that capture the cell phenotype of the cell. And lastly, the availability of electronic health records and the standardization of those help us create a robust data set to start analyzing. Together, of course, with all of these large data sets, we have challenges, such as how do we store those data sets, or we need more computing power now to analyze this large number of data sets. When we analyze, how do we interpret them? How do we integrate these different data types? And of course, we need the coherent data management plan to manage all of these data sets so they don't get lost. And in particular in AI, since this is our industry for groups, it's very important to see that all of those data that we mentioned can go and help and support all the different areas through the medicine. So from the preclinical research, we can use AI methods to find new targets, more efficacious compounds. And in the clinical trials, we can use AI approaches to predict the drug efficacy or maybe stratify patients according to cohorts. In the patient care, AI has also a huge role, especially for predicting patient intake, patient readmission rates, and so on. And lastly, the biggest so far impact of AI comes to diagnostics, where the ability to really process and understand images has seen a tremendous impact in the fields of dermatology, radiology, and pathology, which the AI predictions often match the ones from the experts. After this introduction, now the two main elements of the talk, which is going to be the first part, as I said before, go over a few lessons of learned while the development of this data ecosystem for our cancer center here at the University of Miami. And then afterwards, I'm going to give you a few examples of our research or previews of our work on how we can use now well-annotated data sets for the identification of novel compounds and cancer therapies. A few words about our cancer center. So it's the only cancer NCI designated cancer center in Florida, in South Florida, and one of the two in the whole Florida. And that means that as a National Cancer Institute approved or designated center, we have the ability to use state-of-the-art technologies, approaches, and be very research-oriented. Overall, in our cancer center, we have 97 cancer research laboratories, which are split into 3 multidisciplinary research programs. And of course, the majority of the data generation of all of those labs are being done in the 9 shared resources. The typical pipeline of what happens here at the cancer center is that you have labs, you have research scientists that they do experiments, animal work, cell work, or any other model, and then they submit their experiments, their samples, into one of the 9 Sylvester shared resources. The resources are state-of-the-art facilities, they're cores, and they use state-of-the-art instruments to generate different data types. For example, genome sequencing, RNA sequencing, we have ChIP-seq, ATAC-seq assays. They have an imaging and animal modeling cores to model your experiments in that way. And we've seen that throughout the years, in all shared resources, we have an increased data generation. This particular example is from the oncogenomics, the sequencing shared resource. And over the past 5-6 years, we've seen that more researchers are now sequencing, they're generating more data. And apart from that COVID-related, or infamous COVID-related dip, we have increased our needs for better data management and storage solutions to handle this influx of increased data. A big bottleneck now, once you have an increase in data, is that unless you have a proper data management solution, then it's going to be extremely costly down the road. In a famous research report from the European Union and PwC, it was predicted that the improper data management costs about $25 billion. And it was split between indirect impacts and direct impacts. And the specifics from the direct impacts, and it's something that we see from our experience all the time, they're broken down in this list here, which is the most one is the time spent. So the researcher spends most of their time, the typical 80% to clean and process your data, that it's the saying, and also the cost of storage, the licensing costs. So all of these costs add up and are necessary to be dealt with if we want to have a proper data management system. And how do we do this? So for a big guideline is to follow the FAIR guiding principles of scientific data management. They were established from 2016, and now major organizations, consortia, and institutes adhere by them. And a brief overview from those are that the data that we generate have to be findable, so they have to be searchable by both humans and machines. They have to be accessible, so they have to be stored and accessible in a standardized and secure way. Interoperable is a very big thing, an issue, so the data have to be able to integrate them with other data types and other applications. They cannot be in a proprietary format, for example, and they have to be reusable. When we store the data, we need to store also all the necessary information to reproduce the experiment and not have incomplete parameters. So to go into how to build a data ecosystem, we had to go back and see the overall end-to-end research pipeline here at the university. And this is the typical data-driven research pipeline that applies to all biomedical research. So on the first step, we have a data generation step, and this comes usually from, in our case, from the shared resources. So the samples are being sent to one of the 9 shared resources, and they generate data. The second step is the data processing step and management. All the instruments, of course, are not going to give you process data. They give you raw data, and then using different pipelines and automated approaches, you can arrive to the process data, which you then take into the data modeling step, where bioinformaticians, computational biologists, and software engineers and data scientists can take the data and make, in the end, actionable insights depending on the field. In our group and in our environment, we're usually interested in biomarkers and in finding new compounds and new precision therapies. So having those guidelines, we wanted to build this data ecosystem that's going to be future-proof, and it's going to handle now this extreme influx of new data that have been generated. And of course, a few functionalities and the wish list that we would like to have is ability to store the data securely, to access the data, to process the raw data, have some data exploration functionalities, and also to be able to share the data. And so I think 1.5 years ago, it's been now, we built on top of some other data portals that we have done in a previous consortium. The biggest one was the NIH consortium, and we started developing from the ground up one portal for the Cancer Center. It's called the Sylvester Data Portal, not a very unique name, but and currently it's only accessible under the university's VPN network, but we have plans to make this accessible to other researchers in the future as well outside the firewall. And a few lessons from the development of the portal are split into 4 arms here. First of all, it's the data repository functionalities of the portal and any data ecosystem that we want to handle such data. The data management functionalities are very important. The data analytics of the portal, so it's crucial. We didn't want to have just a glorified Dropbox account, but we also wanted for researchers to be able to view their data, explore, make new hypotheses intuitively with their data set. And of course, as you know, cancer research is very interdisciplinary nowadays. We wanted to make as intuitive and as flawlessly as possible to share all the right data, metadata, and QC files to the right people. So for the data repository, it's not a very unique, but it's a very common use case that we were continuously seeing across the university and across our collaborators in other universities. So the use case goes like this, that you have the samples generated by the lab. It can take months to generate samples, especially if it's mouse work. Then you send the samples to be processed by an instrument, either in a shared resource or some external collaborator. And as you can imagine, you don't get one file from every sample. You get a series of different data files. So you have the metadata of the experiment, the raw data, the QC files, and the process data as examples. And each of these files, in the end, what happened was it was ending up in different storage solutions. So the metadata were usually kept in the lab computer. The raw data, in the case of, for example, RNA sequencing, it gets uploaded to BaseSpace, which is Illumina's cloud solution. The QC files were being transmitted and through an email to some member of the lab. The process data were kept in the postdoc laptop, most of the time, maybe in some lab server. And this was happening for a while and was working well, but after a few years, when it's time that you want to publish your data or to start a clinical trial or to apply for a patent, you need all of these key pieces of information together. And especially, for example, due to postdocs leaving the lab or different staff members leaving or equipment being changed, we were seeing that we were losing data. So the first use case that we tried to solve is let's have a data agnostic portal that we can store every data type in there. It doesn't have to be raw sequencing data or proteomics data. So, and this is the thing that it serves. And of course, building such a type, we need to have a ton of considerations. Three lessons here for just to consider is that we definitely need multiple security tiers. Are your data patient health protected health information or not? You need different access tiers. Are your data types frequently accessed or rarely accessed? And of course, you need to think about the storage location. Do we store them in the cloud? Do we store them on premise and they're more secure? And so on. On the data management aspect, we wanted to build it from the ground up and adhere to the FAIR principles. So everything at store has to be findable, accessible, interoperable, reusable. For this, we built standardized metadata and data specifications. So for every assay that we're starting to incorporate in our data ecosystem, we have experimental metadata, QC metadata, and reagent metadata to really standardize what is going on. And lastly, we want our data sets to be publication ready. So whatever information is needed to publish, say, for example, to submit in GEO your RNA sequencing data, we want to make sure that we capture as much as we can from the first step, from the time the data set has been sequenced. The data analytics aspect of it, as I said again, was we just did not want to create a data repository folder, but we wanted to create something that's alive and helps both computational but also experimental researchers with exploring their data. We have on our backend, we have an API-based R and Python environment that can handle different widgets and tools. A big component of this is the automated processing pipelines. So the moment that the data have been generated by the instrument, we put them into automated processing pipelines to process them immediately and give the option to the researcher if they want to keep this our own pipeline, or maybe they want to use again the raw data for their own analysis. UI-based tools was on the forefront of our development. So we have different more data exploration than data analysis tools. And integration with publicly available data was very important. So we connect with API to more than 10 bioinformatic tools for different pathway analysis, enrichment analysis. The trend now is to have this microservice feature. So it's very nice to be able to connect via APIs to other people's work. And finally, our data, we wanted again to prioritize and make them not only publication ready, but AI ready. So the data come with clean metadata, standardized vocabularies, and their machine readable formats. So the researcher can take it immediately from the portal and give it to the computational biologist for the downstream analysis, and therefore fast track the analysis process. And lastly, the portal is a collaboration hub. And for this, again, the important aspect was how do we transfer all the data to the right people? As you can imagine, we have metadata that come from a LIMS system, the raw and QC data from the sequencers, the process data for the automated pipelines. So we package everything in one, we call it SDP data set that has the standardized metadata, supporting files, everything together. And then we build on top a secure permission authentication system with two entry points, the UI-based and the API-based. This way we tackle and handle both the experimental researchers and also computational researchers for their downstream analysis. And a few things to consider on this system, it was actually the typical that has been doing this in the world is that we have different user roles. You can imagine that maybe the administrators or the people in the shared resources want different widgets, different tools, a different view of the portal. The data sets have a different privacy levels. Some of your data you don't want to share with anyone, or maybe some of them you want to share them within the university, make them public. So it was very important to allow for that. And also different permission levels. Every data set comes with a specific permission for that person. And that really helps when you want to include a lot of people in the analysis. For example, for your undergraduate or your intern, you would only give a read-only permission, so they wouldn't be able to delete by mistake any data sets. So overall, this is the overview and the different functionalities of what happened in the portal. And now I can go over for a few examples of once we're using this very well curated and well managed ecosystem, and we have those standardized data sets, how can we proceed for the analysis aspect? And of course, our ultimate goal again is to find new therapies and new targets for cancer. And this work includes a few examples that from the group that I'm in, it's called Systems Drug Discovery Group, and it's been led by Dr. Stefan Schurer, and also from our collaborations, the NIH Links Consortium and the CPTAC Consortium, which we are members, and working on the analysis aspect of it. So let's see. So 3 examples that I'm going to go over here. The first one was, apart from the Systems Drug Discovery Lab, we have a very good cheminformatics background. And one of the biggest bottlenecks on the drug development pipelines is the time that it takes to take a drug into approval. The typical example is that, as you know, that it takes more than 10 years to reach approval, and also from the 10,000s of small molecule lead compounds that arrive into the drug discovery stage, only one compound eventually gets approved. A big reason for this is the efficacy, of course, of the compounds, but also is that the chemical space, in comparison and in contrast to, for example, the genome space, the chemical space is vast and ginormous. In this figure here, you can see the number of small molecules that you can right now buy from some commercial library. So they reach about almost 4 billion compounds that are right now available. To put it into perspective, it's more than double the number of stars in the Milky Way galaxy. So we're talking of an enormous number of compounds in the chemical space. Also, the chemical space evolves over time and becomes bigger. These are the different chemical libraries that you can right now buy and synthesize compounds for, and you can see the trend that throughout the years from 2015 to 2018, you go a big increase in the number of compounds that you can explore and then screen and then find ideal targets. And of course, to put it into reference, the number of approved drugs is the small list here. And you can see how small it compares to the availability of different compounds. So there is a need here. We have a lot of compounds that we need to screen for activities, but we cannot, of course, test them all experimentally. So the best way is to develop in silico methods to first prioritize and screen all those large chemical spaces. And this is an example that we're basing our work here, some of our work in the lab. This was a preliminary example of how we can use deep learning to screen the activity for chemical activities. Our data set that we started with was the Campbell datasets and the KKB datasets. And overall, we collected 650,000 data points, which were 300,000 compounds. And so these were activities for 342 protein targets. Most of them were kinases targets. The first step is that, and this is what typically done in cheminformatics deep learning, is how can we digitize, make machine readable the chemical structure of the compound. So in this approach, we use the extended connectivity fingerprints, ECFP4, and essentially binarizes it's like a hash function to binarize the chemical structure into zeros and ones. Now that we have a very good input and readable input, we developed a multitask deep learning architecture that on the input was the binarized chemical structure, and in the output was the predictions for the activity for those 342 targets. And there were a lot of interesting aspects in this. One particular aspect was how does it compare with other machine learning algorithms? So you can see here, every dot here is the area under the curve score for the prediction for a particular kinase. Every plot, of course, has 342 dots corresponding to the 342 kinases. And as you can imagine, the data being sparse, means that for some kinases, we had more compounds, more data. For some kinases, we had less data. This gets reflected here on the x-axis. And here's the interesting part, that for kinases that we had a lot of training data, all the models were behaving similarly accurately, except of the name. However, for kinases that we did not have a lot of data, you can see that our multitask deep learning model had even very good accuracies for many of those kinases, in contrast to the random forest, the logistic, and the naive Bayes models, which gives a sense of the importance of the deep learning neural networks, because they can even work with less training data available, and especially in the multitask aspect, that they really can assimilate information from other training sessions for that particular kinase. And apart from the cheminformatics, the biggest focus of our group is finding compounds and targets for specific cancer types. And for this, we're using the connectivity map approach and two different data types. The ones that we described before, we use the baseline profiles, which is we have two more samples, cancer cell lines, and we want to see what's their baseline expression of protein, RNA, their baseline mutations. There's a lot of consortia that generate those data. I have here the most frequently used ones, the large scale ones. And another set of data that we want to integrate now in this analysis is the perturbation response profiles, which again is what happens to the RNA and the phosphoprotein after the treatment. And here I have a few consortia, with the biggest one being the links, the proponent of this data type, that you can access, freely accessible, very large data sets that you can use for their analysis. A few words for the LINKS consortium. It stands for the Library of Network-Based Cellular Signatures. It's funded by the NIH. This year was it's the last year it's been running. And we had many centers taking part of this, and different centers were generating data, analyzing data. We are part of the Data Coordination Integration Center to bring all the different data together. And the rationale for the consortium is this, that you have different multiple perturbations, 20,000 perturbations, that you treated them in 80 cell lines, and you have signatures. What happens to those, to the RNA, the phosphoprotein, and the phenotype of the cell after the treatment of with those perturbations. And all our analysis is based on this rationale of the connectivity map that say that from the baseline profiles, if we have a disease signature that we know, for example, these genes are upregulated, these genes are downregulated, we also have information about what happens to the RNA for using a drug treatment. So we can map, we can connect a drug that reverses, for example, here the blue genes, to the disease, and this is a very robust way to prioritize compounds. The approach has been applied in multiple diseases like binge drinking, schizophrenia, artery disease, and in particular in cancer. And it's based on this L1000 platform. Essentially, it's a high throughput assay that doesn't measure the whole transcriptome, but it measures 978 genes. So the RNA of those landmark genes. And of course, in our work, we found in other works that compounds with the same mechanism of action elicit the similar transcriptional responses. And we can find what genes change for a particular compound or for a particular mechanism of action. And also, in the end, this drives really the point that we need signatures and algorithms to connect this. In the next two examples I'm going to show for this is, and I think we're running out of time, but I'm going to be as short as possible, is the both examples follow the same rationale. On the first aspect, we want to create a disease signature. So how do we do this? We make comparisons on the RNA level, on the protein level, on the phosphoprotein, giving different groupings. So you can compare mutant samples with a particular mutation versus wild type, tumor samples versus normal, some other subtyping samples. And also, we create those perturbation response signatures that from those consortia here. And the end game is to connect them, find the right perturbation that connects to the right disease signature. We applied this in glioblastoma, and we started with 99 glioblastoma samples. As controls, we used 10 brain sample from GTEx, made a complete proteogenomic analysis with all of those 9 different multi-omic assays, and use the mutation status now. So for all of those samples, we knew what mutations for all of those driver genes. And the next step was to create the disease signature. So again, in the connectivity map approaches, we need the disease signature. We created ones for using RNA data and phosphoprotein data. And then we found, using the different connectivity algorithms, different compound prioritization lists. In this example, we used 2 RNA algorithms and 1 phosphoprotein. So we had 2 RNA prioritization lists and 1 phosphoprotein. This is a special example. We did this for multiple contrasts. But in this particular, even if the RNA algorithms were agreeing that HDAC inhibitors would be reversing the RNA signature, we saw the remarkable thing that on the phosphoprotein level, the EGFR inhibitors were showing to reverse the phosphoprotein disease signature. And it's actually very tied to the biology of the EGFR inhibition, which, because it's very upstream, on the RNA level, you have a very low signal. And that leads to the point that the multi-omics assays, using different assay types, such as phosphoprotein, protein, or RNA, you can have different aspects from the tumor biology. Another interesting example was the MEK inhibitors now. So we compared MEK NF1 mutated signatures with wild type. We made, again, 2 prioritization lists using the RNA, 1 of the phosphoprotein. In this now NF1 mutated samples, MEK inhibitors were popping up, they were prioritized in both using the RNA and the phosphoprotein level. And of course, it comes to no surprise, as for example, a few weeks after the analysis, a MEK inhibitor was the first drug approved for neurofibromatosis type 1, NF1 mutated disease. So it was remarkable to see that how we connected, and with real world data too, our findings really indicated that NF1 mutated signatures would benefit from MEK inhibitors, and the real world data also were concluding. And we can do the same approach using different stratifications like immune signatures for RB1. And the last example here was the lung squamous cell. In this, the uniqueness of this is the first step was the same as the glioblastoma sample, like full proteogenomic profiles for every single sample. But the unique thing here was that we did not want to use only mutation data. We wanted to use an unsupervised methods to cluster all the different data types together. So in here, we use the NMF methods to do that. For this, we created 5 multi-omic subtypes. And now, having those robust subtypes, we can create subtype-specific signatures and really gear toward the precision medicine approach. Following the same example again, we have our disease signature, we have our drug signatures, we can make drug prioritization lists. Remarkable one example here was the EMT signature here, that we saw that TGF-beta inhibitors were particularly enriched only in the EMT signature. We did, after collapsing on the mechanism of action, we also saw the same thing. TGF-beta inhibitors were connecting to only that particular signature. And of course, we replicated that using different drug connectivity approaches, and we saw the same thing, that only for the EMT signature, we had TGF-beta inhibitors. And that really was a very good sign and case, and of course, it was validated with the other findings of the paper to connect that particular patient subtype to a particular treatment, and really paving the way of how we can use multi-omics data for clinical decisions. And lastly here, I have a preview just of a few examples of how we can use them in AI. And it's only a few slides, 3 more slides. And one example, this is again the work that we're doing in the CPTA consortium that entails using all of those multi-omics data sets, large-scale data sets, but leveraging now AI approaches. In a quick example that we can do here was that we used transcriptome data and proteome data in a variation autoencoder. Of course, we injected it with external knowledge, and the architecture was a little bit more complex, But the idea behind it was that, and you can apply this everywhere, that you can use these AI approaches to really stratify samples. In this projection here, we stratify them based on cancer type, but looking at different latent variables of their variational encoder, you can find different molecular biology characteristics of the samples. And of course, a remarkable thing was that you can also calculate the contribution of the signal. So in our case, in every model that we've been training, we saw that the protein weights were always higher than the RNA weight. So the protein information was more important for the model to recapitulate again the same signature on the output. And this was also very informative to see what kind and how much information, how do we weigh this different multiomics information to reach to biological relevance. Lastly, in this example here, we wanted to connect now imaging data with different clinical molecular features. And this was work that was already published, but our work continues on this aspect that essentially takes imaging data from our CPTAC consortium and using different levels of viewing fields, we can make different convolutional neural networks that can predict different characteristics of the images. In this slide here is we just see here that using the imaging data only, of course, you can predict the histology of the image, but it was very remarkable that you could predict molecular features, such as the T53 mutation or the P10 mutations. And this is another way how we can join and intersect the different fields, for example, for imaging data with more actionable fields of molecular features, such as specific mutation, and really drive the way of personalized medicine, leveraging all of those AI and machine learning approaches. So overall, just to a quick summary, we went, the first part of the talk entailed the use of how the proper data management is important. And of course, we need to have secure data sharing, long-term data storage, especially important as well, and intuitive data exploration portal for experimental biologists that are not very computation savvy. And also it's very important to have some automated data processing to really get into the hands of the process data of the people right after the instrument is finishing with the sequencing. And on the second part, we saw now that everything after is standardized, well annotated with the proper metadata. It's easy to integrate the data sets, proceed with different data modeling approaches, such as machine learning approaches or bioinformatics approaches. And we saw that we could predict a compound's activity using a deep neural network. And also we could prioritize therapies in glioblastoma and lung squamous cell carcinoma using the connectivity map approaches. And lastly, we saw a brief preview of what we can do now using tapping into state-of-the-art AI architectures to make predictions about the molecular features of the cancer, and also intersecting and joining imaging data with other data types. Overall, the groups I would like to thank is, of course, the System Drug Discovery group that is leading by Dr. Stefan Schur, our department, and my team for the Sylvester data portal, very helpful, the whole Sylvester Center. And of course, it's very important, the collaboration among scientists and the contribution of both the NI Links program, the CPTAC program, has really shaped our research ways. And I think for now, we can have a few minutes for any questions or discussions or whatever is needed.
Thank you, Vasily. Typically, I'll leave other people to start first. But in this case, because I have to really leave in 5 minutes, and I have about 10 different questions, I will just prioritize and ask one or two before I go, and you can continue afterwards. At the same time, I would like Tassos to try to arrange a separate meeting, maybe if it is possible for you, Vasily, to discuss some of the issues, because your work is touching upon many of the things that that we do. One of the things that I'm interested in is to how you use how you standardize metadata. Do you use common ontologies? How fine-grained do these need to be? Who provides the metadata according to these ontologies? All this verification process, which I know it's quite expensive.
Yeah, so for the metadata, you're right. We have two ways. First of all, for our reagent metadata, we built on top of our already developed links metadata, which was a big effort to create standardized fields for all the key reagents. We also, for example, for our gene metadata, we bring in definitions from ontologies. Our cell metadata, we connect with Cellosaurus. Now, the important aspect, I think, for all the metadata was how can you collect everything that's needed when it's time to publish? And for this, we do the groundwork as well. For example, the first assay that we have supporting is the single cell assay. So we went to GEO to see what is necessary and what control vocabularies are being used when you want to submit your GEO entry. Plus, we incorporated the Minsky. So these are the minimum single cell metadata specifications that have been developed through the single cell consortium. So it was more of combining the best practices from both what is required from the regulatory aspect of the NIH GEO, plus also look for other consortia for that particular assay if they have released any specifications or best practices.
So your metadata is specific to the assays that you're working on?
Yeah, so we go assay specific, and of course, on the back end for all reagents, we are incorporating the links metadata, which were developed 3 years ago and to support the whole consortium.
Don't you need also some metadata about the experiments themselves? So if you have different types of animals, you probably need different protocols and different metadata for these.
Yeah, so for experimental metadata, so what was the protocol? We haven't given it, we haven't implemented it yet. For only the experimental aspects that we're incorporating now comes from the shared resources. For example, what was the library to prepare the RNA sequencing, what reagents were used. These ones we collect. But now the true experimental metadata of the specific protocol that was followed, we don't have it, we're not collecting it yet.
Right. This is related also to another question that I will ask, and then I will just leave after that, which is how easy is it for somebody else to use your system? Because to me it would seem ideal if it is open source. I don't know if you intend to make it open source, for somebody else to be able to use the same approach for a different disease, for instance. We work a lot on rare diseases, for instance, which is where this kind of infrastructure is very valuable. Would it be possible at all?
So there are two aspects on the system, right? Is the software development aspect, which is just what particular software and stacks we're using for development. I don't know if this benefits because technology changes. For example, we're using Azure, someone else is using AWS. But on the data management aspect, I think we're aiming, of course, to publish and make it available, or our specifications of how we integrated, what control vocabularies or ontologies were used in what particular, because as you said, you don't have only reagent specifications, you don't have only QC specifications, but it's a whole group of different metadata specifications that need to be coordinated to fully describe an experiment. But yeah, on that aspect, I think that would be the goal, to make a release either as a white paper or as a publication. Essentially, it would be something like best practices or insights of how can we correctly, appropriately manage our data.
Okay, thank you very much. I will really have to jump to another meeting. I would like to ask everybody to continue the discussion in English if possible, because I would like to share the video with some non-Greek speaking people. So again, Vasily, thank you very much, and I'm looking forward to our meeting to discuss more things.
100%. Thank you. Very nice meeting you.
Thanks. Bye-bye.
Bye. Goodbye.
So I think that Fotis has a question.
Yeah, of course, of course.
Yes, hello. Actually, I have tons of questions, not only one question, as like George, because we're really doing similar stuff. I mean, it's very, very interesting for us. Thank you for the interesting presentation. We have worked a lot on similar research tasks in this area. But of course with a different experiment structure. So I'm going to just mention a couple of questions myself as well, because I don't want to hijack the whole presentation, the whole session, and also need to leave it in a while. And we should definitely, I think, if you Vasily is available, we should definitely have another more focused call. So thanks. So just initially, as basic question, we have done also personalized the medicine approaches for lung cancer and for rare diseases, like George mentioned, and Alzheimer's. What we did in meaning when we have different experiment structure is that we have used, for example, in tasks like drug discovery, we have combined many kinds of disparate data in a single database. So as far as I understand, you collect genes and targets and information related to clinical records, but you don't combine them to have conclusions based on everything. So we have what we actually have done is that we have tried to semantify them and link different data sets and disparate data, like not only genes or targets, and analyze them together. So just the question is if you have considered working on something like that, so combining in your analysis different kinds of data.
No. So yeah, for us, like you said, it was more straightforward. Usually the input is some kind of a gene expression profile, or sometimes we have merging layers. So the disease gene expression profile plus the perturbation, the drug profile, drug response profile. But yeah, there are definitely, and there are many, I think Benevolent AI also is focusing on that, creating AI models that have disparate information. And I think it's very valuable. One thing that we saw was, for example, when you interject in the architecture of the model external knowledge, say it's a pathway, right? That instead of your genes, you have a dense network, maybe you have a sparse network that connects the right pathway, you really increase the accuracy of the model as well. So that's one lesson, but also, yeah, we haven't tried to connect disparate data into our training.
Okay, thank you. And another quick one, because I see people are less, so somebody else may need to ask something. I had a real trouble working such tasks about drug discovery, for example, in mapping the same drugs in different databases. I saw that you are using KMBL, for example, and KKB, I think. So have you had similar issues, and how did you overcome mapping, for example, drugs from drug bank or from KMBL or around different databases into the same entity?
Yeah, that's actually a big question. It was a big bottleneck in the consortium. What we did was we found a solution, but only for the 40,000 compounds that we used in the consortium. And essentially, we had to build from the scratch standardization pipelines for the molecules. Like whatever it's out there, it's super dirty. Like some smiles have the salts on them, some of them not. You can have two smiles that are isomers and they can reflect different chemical IDs. So yeah, it's a lot of work. And for us, for the consortium, like we had to build it from the ground up and build a pipeline that says, okay, these two chemical IDs are essentially the same. We call them links, small molecule IDs. Or not even in chemical, right? You go to public data sets or they only use the name of the drug. Good luck with that. Like if they only use the name, it's but
Exactly.
there are multiple names. There is a big disjoint in the annotation of small molecules. Maybe
Just for your information, in case you're interested, I have, because I struggled a lot with this, and I'm finishing that, I have tried to aggregate from 6-7 databases like DrugBank, UMLS, KMBL, PubChem, etc., all the IDs, and trying to map them using any kind of linking I found in this database, and I have uploaded them in a GitHub public file, so I could provide you, if it's helpful for you, I could provide you a link with that. And we would be also interested for data sets that you provide, like, for example, the targets that are found for lung cancer or the drugs that are found for targets. So we would also...
I think the connection is...
And we can follow with this offline. Thank you.
No, 100%. And like during my PhD, that was a lot part of my work. Like go Drug Central, Drug Bank, Campbell, and somewhere I have like notes of what inconsistencies and where all of those mappings were failing. It's a very important work. That we're happy to connect after.
Okay, thank you.
So I see that no other hands are raised. Of course, more questions exist, but I'm afraid that the timing, we are already 7 minutes late, and perhaps also technically our call may be shut down. So I will contact you, Vasily, to arrange another call so that we can have a discussion. Again, thank you very much for this very interesting discussion, and we will collect our questions so that we can discuss them.
Perfect. Thank you, and glad to meet everyone. Take care. Have a great day.
Bye.
Bye, guys.
Bye.
Thank you. Bye-bye.